person.
Yes. Yeah.
I wanted to see more than one person.
So it's my pleasure to introduce today's Socaar seminar speaker, Rachel O'Brien, who's an assistant professor at William & Mary. Rachel got her BA in Grinnell College. She did her PhD at UC Berkeley, and then went on and did postdocs at Lawrence Berkeley National Lab and at MIT. And in her PhD and in her postdoctoral positions, she's been studying different aspects of atmospheric aerosols. So she's a great speaker for Socaar. In her position at William & Mary, which she started in 2017, she's been leading a group of undergraduate students and master's students studying various aspects of indoor air chemistry and aerosol photolysis. So with that, I will encourage everyone who's not Rachel to mute their microphone and hand the floor over to Rachel.
Thank you so much for the introduction, Jen. And thank you also for the opportunity to speak with you all here today. So today I'm going to be talking about some of the work that we're doing, looking at the photolysis of atmospheric organic aerosol particles. I'll be looking at chemical transformations as well as photobleaching and sort of changes in the optical properties. Before I get started with that, though, I want to give a little introduction to my group. So as Jen mentioned, we are a primarily undergraduate department. That means the majority of the students in my group are undergraduates with a few master's students. And there's a pretty big range of different projects we're working on, and I don't really have time to talk about all of them today. So I just want to give a little shout out at the beginning to some of the other projects that these great students are working on. So some of my students are working on indoor surfaces and really looking at the chemical composition of this surface grime that can build up. I've got another group of students that are working with me on a technique that allows us to look at action IR spectroscopy in the fingerprint region for molecules or ions in a mass spectrometer. And then finally, there's a bigger group of students that are kind of moving outdoors with me. We are looking at the composition of material on outdoor surfaces, as well as we will be looking at outdoor organic aerosol particles. So organic aerosols are kind of a major theme of a lot of the research that I'm doing. And a brief sort of introduction or overview to this, organic aerosol particles are solid or liquid particles that are suspended in the atmosphere. And there's a range of different sources for these. We have volatile organic compounds. These can come from biogenic or anthropogenic sources. And these can be oxidized in the atmosphere. When they're oxidized, their vapor pressure is going to lower, and they can either condense onto pre-existing particles or they can form new particles. And we call all of this secondary organic aerosol because it's formed in the atmosphere. Some of the other material that we look at is stuff that is going to be directly emitted. So this is primary emissions or primary organic aerosol. And biomass burning is one of the major sources of that that we are looking at. So for organic aerosol particles, there's kind of a couple main areas that we think about or that we're concerned with. One of those is their health impacts. In general, there's pretty clear indications that higher concentrations of these particles in the air leads to negative health outcomes on a range of different levels. And then the other side of this, and what I'm going to be focusing on in this talk, is thinking about the climate impacts. So when we're thinking about climate impacts for organic aerosol particles, we're really talking about radiative forcing or radiative effects. And so this is an older figure, the IPCC report from 2013, and it's showing the total radiative forcing we have for all of the short and the longer term forcing agents that we have in the atmosphere. Up at the top, we've got our greenhouse gases, and you can see that these are kind of net leading to global warming, as we know. We can also see that the error bars on these are relatively low, so we've got some good understanding of the amount of radiative effect these have in present day compared to pre-industrial times. What we have in the middle, though, is the region that I'm focusing on, and these are our aerosol particles. So there's two main climate impacts. There's the direct effect as well as the indirect effect. For the direct effect, these particles can either directly absorb or scatter solar radiation. And so net, the majority of the particles are acting to scatter, so they have a cooling impact on the climate. There's a subset of these that are dark colored. These are black carbon or soot particles, and they can contribute to net warming. The other one of these, or the other side of this, is the fact that these aerosol particles can act as cloud condensation nuclei. So this is the indirect effect. And this is, again, leading to net cooling on the climate. So overall, these aerosol particles are helping to counterbalance some of the warming that we have coming from these greenhouse gases. But we have some really big error bars on these measurements. And so one of the targets that we have is to really try to better constrain or better understand the radiative effects that these particles have in the climate. From an organic aerosol standpoint, one of the main questions we have is just how much of this material do we have up in the atmosphere? So I talked a little bit at the beginning about where these things are coming from, our formation in the atmosphere or direct emission. The other side of that equation is talking about the deposition or the removal pathways. Now, we can have deposition that is either wet or dry deposition, and the lifetime for that is something like 1 to 2 weeks in the atmosphere. So these are physical processes removing the particles. The other loss mechanism that we don't have as good of an understanding or as good of understanding of the rates for, and those are the chemical loss processes. The way that this is working is if you have a chemical reaction that causes a fragmentation to occur in some of this organic material, if you have a volatile fragment that's formed, that can then volatilize, and the total amount of organic particulate mass that you have will then decrease. So there's 3 main types of aging that we talk about for these particles. We talk about heterogeneous and aqueous phase oxidation. For both of those, it's kind of the same idea. We have an oxidant coming in, something like OH radicals or ozone, and these are acting on the chemicals in these particles. And if you happen to have an oxidation event that leads to a volatile fragment, you're going to see a decrease in mass. The one that we focus on in my group is photolysis. For photolysis, we have a photon coming in, and if this photon can be absorbed by the organic molecule, that organic molecule will be excited up into an excited electronic state. And then it's going to relax back down to the ground state. And depending on the path that it follows, it could lead to a fragmentation event, which then, depending on the volatility, could lead to a decrease in the amount of mass that we have for these particles. That leads me to the overall outline that I have for the talk that I'm going to be giving today. One of the main things that I'm thinking about in my laboratory is really trying to understand the gap that we have between our laboratory measurements and then what we have from ambient data sets or from modeling studies. So I'll be focusing on 2 different studies. The first one is going to be looking at biogenic secondary organic aerosol, and I'll be focusing on mass loss that is occurring when photolysis. The other one that I'm going to be focusing on is biomass burning organic aerosol. And for that one, there'll be a little bit of shift, and we'll be looking at color loss as a mechanism for thinking about the change. And then at the end, I'll kind of bring this together and talk about the remaining gaps and the next steps that I think we want to target to better understand these questions. All right, so thinking about this mass loss and this biogenic secondary organic aerosol, the motivation for this is coming from an older paper. This is Colette Heald's paper in 2011. In this paper, she did a modeling study and then a comparison between the amount of organic that she was modeling in the atmosphere relative to what was measured for some aircraft campaigns. And they found that there was basically a gap between the models and the measurements. And when they adjusted the model to match the concentrations down at the surface, which is generally where we have more measurements, they ended up with too much organic aerosol mass up in the upper troposphere. Now, there's been a number of previous studies that have been done that have really tried to target or understand this gap. A recent one that's come out is the Hodzic et al. Paper. This is in 2016. In this paper, it's another modeling study, and what they did to try to close this gap is they introduced some of these chemical loss mechanisms into the system to try to remove some of this lofted organic aerosol mass. And so one of the main things that they added was photolysis. And when they added this photolysis in, they saw a better improvement, right, in terms of the overlap. But this really raised the question for me as to whether or not we wanted to make the assumption that this type of aging and this extent of mass loss was appropriate for the type of mechanisms that we have in the atmosphere. So this question is coming from some of the work that I did as a postdoc. So this was at MIT and Jesse Kroll's lab. And this is work that was done in the environmental chamber there. This is a 7.5 cubic meter Teflon bag. It is temperature controlled, and then I was running these experiments at a low relative humidity. For these experiments, I was generating secondary organic aerosol from alpha pinene, so a biogenic source, mixed with ozone, so ozonolysis. And I was condensing these down onto ammonium sulfate seeds. And then I can measure these with an aerosol mass spectrometer, and I can track the amount of organic material that we have relative to the sulfate. So I get this set up and I generate these seeds, and then I turn on these banks of UVA lamps. And when we turn on the UVA lamps, what we see is a decrease in the amount of mass that we have for the organic fraction relative to the sulfate as a function of time. Now, this is not a new type of experiment. There's actually been numerous experiments that have been carried out to look at this type of decay. What I did that was new here is that I tried to look at this for as long of a period of time as I could in this chamber. So when you run a chamber experiment, you're continuously losing the particles. They're depositing out on the walls of the particle, or on the walls of the chamber, and they're also being lost to sampling in the instruments. So for these experiments, I was getting out to 40 hours, almost made 2 days, so trying to make it out to 2 days. And when we did this, what I found was that instead of assuming that all of this material was going to decay, I needed to apply an offset, and a rather substantial offset to the exponential decay that we were seeing. So something like 70%, 80% of this mass was not showing a decrease when we did these photolysis experiments in the chamber. Now, this is a really big shift compared to how we have previously thought of photolysis of this type of material. Generally, what's done for these experiments is you run a chamber experiment. It's like 5 hours, 8 hours long, and you look at the first order loss rate for that, and then you apply that kinetic loss to all of the particle mass that you have in the system. All right, and that allows you to calculate the lifetime that you would expect to have for this material in the atmosphere. So this is a demonstration of what this would look like, and just to talk you through this figure, I'm going to be showing a bunch of different figures that are kind of this style. On the y-axis, I'll plot whatever it is that I'm comparing, normalized. So this is, in this case, the organic fraction that's remaining. And then on the x-axis, I'm going to be scaling these out to an atmospheric time. And the atmospheric time, I'm going to be scaling these to the light that is used in Hodzik et al. So that's kind of our basis for intercomparison between all of these different photolysis experiments. So when I do this for previous data sets, we can see there's a pretty big range in terms of the rates that had been previously observed. Henry et al. And Wong et al., some of the earlier ones were very rapid. Some later ones, Maleka et al., the Hodzik et al. Is actually a modeling study. We're quite a bit slower. And if we apply the same idea to the data set we had, we were kind of in the middle there. But if we take this next step and recognize that not all of this material may be photolabile, what we find is that we want to actually draw this a little bit differently. And what we find is that we want to think of this as an initial decay that's happening over a very short period of time. So something like 1 day, maybe 2 days in the atmosphere. And then for the rest of the time, this material doesn't appear to be decaying, right? It is stable relative to direct photolysis. So we're calling this photorecalcitrant secondary organic aerosol. It's material that cannot decay due to direct photolysis. So on a mass basis, what that means is that some fraction of this mass is photolabile. A fragmentation event will allow it to volatilize into the chamber, but the majority of this material is really staying behind and is stuck down on these particles under the aging conditions that we are looking at. So this idea of this photorecalcitrant material is something that I really wanted to probe a little bit more during my career. So that is something that we have been targeting in my group at William & Mary. But we're taking a slightly different approach to the setup and to these experiments. And we're doing this because of that time frame limitation that we have for how long these aging experiments can be carried out, right? If we run these in a chamber, we're limited to how long we can keep those aerosol particles lofted in the chamber. So to get around that, we're actually just collecting our material on a filter. And then we are shining the light directly on the filter. And we have this filter set in a closed box, and we are continuously flushing that with zero air. So it's a continuously flushed box. We have a hole cut in the front, and then we have some Teflon film taped across the front. So that allows us to get the light shining directly on these filters. We can also do some of our aging experiments in kind of the more standard method, which is to do an aqueous extraction and then put them in a quartz cuvette and do a bulk aqueous phase photolysis for the material. Over on the right, I'm showing you the spectrum that we have for our xenon arc lamp in blue relative to what we have for ambient solar. You can see that we have a pretty similar shape compared to what's in ambient solar, but we are about 3 times weaker than the solar that was used in Hodzik et al. And also because we're using filters, we have a pretty big range of different techniques that we can apply to these to get more chemical detail on what's happening in the sample. So we have a UV vis and ATR, FTIR that we can look at. We can also take this and look at it by re-nebulizing it into aerosol mass spectrometer. So I'll call this offline aerosol mass spectrometry. And we have an Orbitrap mass spectrometer as well that we can use to look at these data sets. So what we did is we took some alpha pinene, secondary organic aerosol from ozonolysis. We put that on some filters and we irradiated it. And we looked at this with all these different platforms over about 4 days of aging in our chamber. Over on the left is looking at that for the UV vis. So this is plotting the mass absorption cross-section or how well this is absorbing the UV light as a function of wavelength. And you can see this is a time series with the color coding. What we see initially is a peak that we would expect to see at 280. So this is carbonyls. There's a lot of carbonyl compounds that are formed during this ozonolysis. But as we irradiated this, those carbonyls dropped relatively rapidly. And at days 3 and 4, there's very little change left in that system. And we see a similar story if we look at this with offline aerosol mass spectrometry. You can see a snapshot of that over in the upper right. And the two main peaks that I want to draw your eye to are the C2H30 plus and the CO2 plus ions. The C2H30 plus, there's numerous different sources for that, but carbonyls are going to be a big contribution. And CO2 plus is going to be largely coming from acids. So what this looks like is something very similar to what we see with the UV vis, a decrease in things that are carbonyl based. And then in this case, we see an increase in our products are likely made of acids. And we can look at this in terms of the time series that's shown down below. And we see a similar trend there as well. So a decrease in this C2H30 plus in the green out to 2 days. But then after 2 days, things are really slowing down. They're stopping. So on days 3 and 4, there's much less change that's occurring in terms of the chemical composition of these samples. So taken together, this gives us a little bit more information on what's actually happening in terms of this photorecalcitrant material. The chromophores, or the chemical functional groups that we have that can actually absorb the UV light, are likely carbonyls, probably some peroxides. These are essentially running out. We photolyze them over 1 to 2 days. And once we have photolyzed those, we generate some acids, but the acids cannot absorb this light. And so we are generating a sample that just is not able to absorb UV light in the wavelength ranges that we have down in the troposphere. And so this is what we can think of as photorecalcitrant SOA. Now, because we are looking at these on filter samples, we can look at some different perspectives of this type of material. And I think this is important to do because if this is a large fraction of the material that we actually have in the atmosphere, right, it only takes a couple of days to form this type of material. This may actually be a significant portion of the organic aerosol that we have in the atmosphere. So one of the perspectives that we've looked at for this is kind of on a molecular level, trying to understand what this material looks like in terms of its molecular composition. So for this, we are using the Orbitrap mass spectrometer, and these are 2 different sample sets from that. Up top is a comparison between the dark and the photolyzed sample for 1 day of aging, and then down below that is what we see after 4 days of aging. In the first one, after 1 day of aging, we see what's actually been measured before, some small shifts in the overall composition, but they're generally pretty similar to each other. So the Orbitrap is not a really good reporter for the type of chemistry that we see from photolysis. But then after 4 days of aging, now we start to see some real shifts occurring in this mixture. And what we're seeing, you can really see around, if you look at 500 atomic mass units, this is a big increase in the peak intensity that we have in that region. So we're forming some new compounds in this mixture after it's been sitting there for 4 days. Now we can get a kind of perspective of the time series of this if we look at this in terms of the chemical changes that would occur. So this is plotting the carbon oxidation state, which you can kind of think of as the amount of oxygen that's been incorporated into the system. And the carbon number is then on the x-axis. The data sets that we're collecting for this study are the darker colored ones, and the circles are showing you the photolyzed samples. So initially we're seeing a rapid increase in the carbon oxidation state, but then we run out of our carbonyls, and that chemistry turns off. And instead we shift over to a different type of chemistry that's generating larger molecules on average. And this is likely or possibly oligomerization reactions that could be occurring. I just want to highlight this, though. This is very different from what's been observed previously for photolysis. So the majority of photolysis studies that have been done that have characterized the chemical composition of these mixtures have been done in aqueous, bulk aqueous phase solutions. And you can see the faded lines in the background are showing you what the general trends that we see there. There is oxidation that's also occurring, but in general, things are getting smaller when you have them in aqueous solution. So this is a very different type of aging and a different type of composition that we would have if we were thinking about this photolysis happening for dry aerosol particles, as opposed to what we'd expect to have for cloud droplets. All right, so that's kind of one perspective of this photorecalcitrant SOA, kind of what it looks like. Another perspective is how is this going to behave in the atmosphere? And specifically what I was curious about was how it would behave if this was actually incorporated or uptaken into cloud droplets. Look at this, what we did is we used a tracer for photolysis. In this case, we used 4-nitrophenol. 4-nitrophenol is a nice brown carbon molecule. It's got a very clear absorption at around 325 nanometers. And we can watch its decay as a function of time, in this case in bulk aqueous solution, so trying to represent cloud water. So we ran this with 4-nitrophenol, and then we mixed in some fresh secondary organic aerosol, and then we also mixed in some bleached secondary organic aerosol. And we intercompared between the rates that we see for these different samples. What we see for brown carbon alone, 4-nitrophenol, is a rate that is okay. It's a little bit on the slow side, a lifetime of 3.5 hours in front of our lamp. When we mixed in fresh SOA, it went a little bit faster, which is what we would expect. John Abbott has actually shown that when you make alpha-pinene SOA and you photolyze it in an aqueous solution, you generate OH radicals. And these OH radicals can then also act on this brown carbon, and they will make it photodegrade potentially faster than the original. When we mixed in the bleached SOA, then we saw something different. When we put in this bleached SOA, we saw it slow down, and it slowed down even beyond what we had for the brown carbon alone. What this suggests is that this brown carbon is itself producing some secondary products, right? Some OH radicals that can help degrade itself. And these are being scavenged by this bleached SOA. So this has a kind of different perspective. If we think about this photorecalcitrant SOA, it has a different type of chemical composition compared to what we think of for photolyzed material. And it also, depending on the concentration of it that we have, could play a role in changing the rates of decay that we have for some of the other material that we have, especially in things like aqueous solutions and like in cloud droplets. All right, so putting those ideas together, I've been focusing on biogenic secondary organic aerosol particles. And what we're seeing is that a large fraction of this material appears to be photorecalcitrant, and we just can't get it to photodegrade in front of the lamp through direct photolysis. And this really raises the question then, should we be applying a full photodecay to this as they're doing similar to the Hodzic et al. Paper, or is that an overestimate? And from the perspective of biogenic SOA, I would say that it looks like that's an overestimate, right? We wouldn't want to try to photodecay all of this material. Some of it is just not photolabile in the atmosphere. All right, so now a little bit of a shift in perspective. We're going to go to the other type of study. So this is looking at biomass burning organic aerosol particles. So for biogenic SOA, these are generally kind of white or transparent material. They are kind of scattering. They can absorb light, but they're not doing a lot of absorption. For biomass burning, it's a different story. Now we're in brown carbon. There's a lot of light absorption that can occur. The first demonstration of this was in 2004 in Kirchstetter et al. What they did is they collected some smoke on some filter samples, and they compared the amount of light attenuation or absorption that you have relative to what they expected to see for bare soot particles. So that black bare line down at the bottom is bare soot particles, and then you can see there's absorption above that. What they see is increased absorption, especially at shorter wavelengths, and they also found that this material was soluble in acetone, right, suggesting that this material is organic molecules that are contributing to this absorption. Well, we now know quite a bit more about this type of material. We know the chemical structures tend to be they tend to have conjugation in them. Aromatic compounds and polycyclic aromatic compounds are common. There also can be substituent groups on there and functionalization. All of that is contributing to get the absorption for these molecules down into the visible region, which gives them a little bit of a yellow color or even a darker brown color when we look at them. Now, the color of these particles is really important because they can actually play a role in the direct radiative effect. So I talked a little bit ago about how most of the particles that we have in the atmosphere are doing some scattering, but the black carbon are doing some absorption. These brown carbon molecules can likely also do some of this direct absorption, and those aren't currently included in the IPCC estimates for the radiative forcing. So there's some uncertainty there. But this is also chemically reactive. And as these molecules bleach, they're going to go from absorbing, or a brown color, to a transparent or a white color, in which case, depending on the composition of the particle, this could switch from an absorbing to a scattering type of particle. And so understanding the lifetime for this photodecay or this photobleaching is kind of a major concern for really trying to understand and quantify these radiative effects. Now, there's been a pretty big range of different laboratory studies that have been done to try to look at this change. And this is just a snapshot of some of the, I think, prettier figures that I have seen. In general, the summary from the laboratory studies is that there's a lifetime for photodecay that's relatively fast, something like 1 to 5 hours. More recently, there's also been some reports for some slower lifetimes, multiple days out to multiple weeks. And this is the Fleming et al. Paper from 2019. A lot of these are also reporting some residual brown carbon. I'm going to be calling this photoresistant brown carbon. So this is material that is able to absorb light, right, is able to absorb light that is in the troposphere, but it's maintaining its color for a long time. It is resisting photobleaching in the atmosphere. So this is what we have from the laboratory side. I want to focus a little bit more detail on what we have for the ambient side. For ambient studies, the first one of these was Forrester et al. In 2015. Here they're looking at the amount of brown carbon and they're normalizing that to carbon monoxide. And they found a lifetime of about 12 hours in the atmosphere. The subsequent year, Wang et al. Came out with a paper. This is from fires down in the Amazon. And the lifetime here is a little bit more complicated, so I'll get to that in a minute. Next year, there was one that came out, Di Lorenzo et al. Here they were not looking at the kinetics. They were looking at kind of a single time point set, but they noted that the decay was very similar in terms of the transport time to what was observed for Forrester et al. And then more recently, there's been a paper that's come out. This is the Socaar et al. In 2019. So this is biomass burning coming out of India. And here they found a much slower decay. So this is a lifetime of about 4 days in the atmosphere. So for the ambient data sets, we've got a pretty big spread in terms of the lifetimes, something like 12 hours out to 4 days. And there's also some level of consistency that there is residual brown carbon or this photoresistant brown carbon that's being observed in numerous samples. All right, so understanding this gap that we have in terms of the lifetimes is one of the targets for the studies we are doing. For these studies, we are looking at Firex filter samples. So there's 2 different sets. The first set was collected in 2016 at the Missoula Fire Lab, and then the other set is collected during the Firex AQ experiments in 2019. And we're doing 2 different types of aging. For the filter aging experiments, because we only have 1 filter per sample, we are slicing these into quarters, and then we are irradiating each quarter for different periods of time. For the aqueous phase experiments, we're just extracting this into a bulk aqueous solution and then doing the photolysis for those experiments. Now, before I get into the first of these studies, I want to talk a little bit more about the method that we are using for this data analysis. And this is something that is a little bit challenging when we're trying to talk about brown carbon bleaching, right? I showed you at the beginning what we had for alpha pinene SOA. It had that really nice clean peak at 280. It's just the carbonyls contributing. When you have a complex mixture of chromophores like we have in these biomass burning or these brown carbon samples, it's a much more complicated spectrum because there's often a lot of different types of chromophores that are present in these samples. And then when we do photolysis, it becomes even more complicated because there's different photo bleaching rates for the different chromophores. And some of these chromophores are actually going to form products that are themselves possibly able to absorb, possibly at a different wavelength. What that ends up happening then is that the shape of the overall absorption spectrum can vary as you run these photolysis experiments. So there's a paper, Ionia al. in 2018 that I think demonstrated this just beautifully. They looked at benzene SOA under high NOx conditions, and they looked at the photolysis. And you can see from the difference plot over on the right that there's real shifts that are occurring in terms of the shape. And the challenge here then is what wavelength do you use to define your kinetics? Because if you have shape changes that are occurring as you're doing this photolysis, you're going to have potentially different kinetics that you're pulling out. This is shown in that figure that they have down in the lower left. Now, this is a challenge for a mixture of different samples. If you're looking at one sample, it's not too hard, but we're looking at biomass burning samples from different types of burns, from different fires. And so we have a big mixture in terms of the composition of this material. So to get around this, what I'm doing is something that I'm calling a maximum direct absorption efficiency calculation. For this, I'm taking the absorption that we have for the brown carbon. In this case, I'm plotting it as the bulk mass absorption cross-section. And then I'm overlaying that with a solar flux. In this case, plotting that in watts per meter squared per nanometer. And if we look at the intersection of these two, you can see that we have some overlap. This is a brown carbon, so there's actually quite a bit of overlap. We can plot this as the MDAE in the watts per gram per nanometer. So this tells us the maximum amount of essentially power that this material could be absorbing if it was in the atmosphere. Now, this is obviously not something that we want to extrapolate directly because this is completely ignoring any scattering that could occur. So this is just the maximum amount that you would expect to be able to possibly absorb. What we can then do is look at this and the total area under this curve, and we can look at that as this material is decaying. And this gives us a way to look at a more holistic view of the amount of brown carbon that we have in these samples and kind of look at the photodecay of this material as a whole. All right, so this is the type of analysis that I'll be applying. So looking at these aqueous extracts, this is a set of samples that we have. In red, I have the FireX 2016, and in blue, I have the samples from FireX 2019. Up on the upper left, you can see our starting conditions, and you can see that we have some features, some humps in those spectra, which is strongly suggesting that we are going to have these types of shifts occurring in these samples that necessitate this type of analysis. For these decays, we are looking at dilute aqueous solutions. We are running them at a low pH, so I'm using hydrochloric acid to bring the pH down to about 4 at the start. And then I'm using a concentrated mannitol in there that I'm adding in as a scavenger. So I'm really trying to target direct photolysis, and I'm trying to scavenge any OH radicals or any excited triplet states that are possibly happening in these mixtures. When we look at the decay, you can see the data set over on the right. We are looking at these for extended periods of time in the laboratory. So these are very long experiments. When we look at these, we find that we need to use bi-exponential fits with an offset. And I'm kind of highlighting the region that we have for this bi-exponential. The first of these rates is relatively fast, and this is now scaled to the atmosphere. It's got an atmospheric lifetime of something like a few hours. But it's only hitting a small fraction of the color. So the gray dotted line is kind of highlighting the region for that decay. The rest of this material appears to be more photoresistant, and it's coming down at a much more a slower rate. It's got a lifetime of anywhere from 8 hours out to multiple days in the atmosphere. So a large portion of the brown carbon that we have in these FireX filter samples appears to be photoresistant. All right, so that's what we have for the laboratory. And there's some comparisons and some overlap with what's been seen previously. Now I want to step back to what we had for these ambient data sets. And I'm going to put them all on the same scale so that we can make a comparison. So this is plotting the brown carbon signal that's remaining as a function of the atmospheric time scaled to days. And for each of these data sets, the darker portion, or the solid portion of the line, is showing the portion that is actually coming from the literature itself. And then the dotted line, that's the section that is what I am extrapolating then beyond what was actually published. So in green, we have the Forrester et al. Data set, and that one is decaying relatively rapidly. It had a lifetime of about 12 days. I've also overlaid on top of that a couple of data sets from some single time point measurements. So Di Lorenzo, I mentioned that a little bit ago. There's also a newer one, Zhang et al. Did a study out in the Azores looking at Canadian wildfires. And what they mentioned was that 2 weeks they didn't have any brown carbon. So I've got that labeled at 0 out at 2 weeks. The other ones that we have for comparison is Wang et al. So these are the data sets that were collected down in the Amazon, and they're a little bit complicated because instead of looking at this as total atmospheric time, what they looked at was a photochemical clock. So we have to make some assumption as to the concentration of OH radicals that were present in the atmosphere at that time. So that's why there's two different colors. Those are just kind of two different reasonable expectations for OH radical concentrations. The other things that I'm doing to the Wang data set is it's relatively short decay, and there was quite a bit of signal, brown carbon signal that was remaining after that period of time. And so I'm applying a heterogeneous oxidation decay onto this data set as well, under the assumption that if these particles are in the atmosphere for 2 weeks, they will likely also have heterogeneous oxidation that is occurring over that time period. Now, there's been some recent studies, those are down in the bottom right corner, that have looked at this. I'm picking the faster of these rates just to try to get one of the bounds for what we would expect. And so that's how we can have that continued decay for the Wang data set. And then the last data set is the blue one. This is the Desari et al. Data set, and this is decaying again with a lifetime of about 4 days in the atmosphere. And they had a relatively long experimental set for that experiment. Now I can take these ambient data sets. If we overlay on top of that the laboratory data that we have, what we find is the laboratory data is falling kind of in the middle region between these. This is the laboratory data for the direct photolysis, and then similarly to the Wang et al. Data set, I'm also including the heterogeneous oxidation decay on top of that, because we can assume that if these are in the atmosphere, they can also be experiencing heterogeneous oxidation. So in general, for many of these fires, what we are seeing is some organic material that has brown carbon color out past a week, even a week and a half in the atmosphere. So we have some photoresistant brown carbon. We also have a pretty big spread in terms of these rates still, right? Anywhere from 1 hour out to, or 1 day out to 4 days of lifetime for this material. So there's still a pretty big gap in terms of our understanding for why we have this big of a spread in terms of the photodecay rates for this type of material in the atmosphere. So that's the last part of this talk, is to really look at the perspectives for these remaining gaps that we have for photolysis of organic aerosol particles. To look at this, I'm going to shift to the other type of study that we are doing for these FireX samples, and this is looking at these aged on the filters directly. So looking at these filter samples, we can use different techniques to probe these. One of the ones that we're looking at is ATR FTIR, and one of the data sets for this is shown over on the left. In general, we're seeing a couple of main shifts happening in the chemical composition. We see a rapid decrease in nitroaromatics, and this is pretty consistent with previous work, especially Fleming et al. They showed that some of the smaller nitroaromatics actually had very short lifetimes, and so we're decaying very quickly. The other thing that we're seeing, though, is a big increase in the amount of oxygen. So you can see this as the increase in the C double bond O, as well as in the OH stretching regions. We can say this even more quantitatively if we take these samples and we atomize them directly into an aerosol mass spectrometer. I'm going to go back to this perspective on the carbon oxidation state, and this is looking at this now scaled to atmospheric age. And for comparison, I'll look at the alpha pinene SOA. So this is that first data set that I showed you. Again, we see a rapid increase, but it's only happening over the first couple of days. And it's then settling out, steadying out at longer time periods. If we overlay on top of this what we can see for biomass burning, what we see is a very big shift up in terms of the amount of oxygen that's getting incorporated compared to what we had for biogenic SOA. Now, we have different types of fires during these experiments, and we see differences in terms of the rate and the extent of the oxygen that's getting incorporated for these different types of fires. But in general, we're seeing a lot of oxygen. And we can actually make a comparison to the same types of fires. So this is work that Chris Lim carried out during the FireX 2016 campaign. And this is looking at the smoke going into a small chamber, and then he was doing OH radical oxidation for the smoke mixture. And the amount of oxygen that they saw, the increase in the carbon oxidation state, is more than what we are seeing, but it's comparable to the amount of oxygen that we are seeing increase. And I'll remind you, for our photolysis box, we just have a xenon arc lamp. There's no OH radicals or ozone in the system. So we're seeing photooxygenation that is fairly comparable to what we can see for heterogeneous oxidation. This is surprising to me, and it raises the question whether what we're seeing is just the brown carbon molecules, which we don't necessarily expect to be a huge fraction of the mass that are actually getting oxidized, or whether there's other processes that are occurring in the system. We have some evidence for the other processes too, if we look at this in terms of some of the tracers. So this is the levoglucosan tracer signal. This is M/Z60 or the C2H4O2. And it's generally a tracer for sugars like levoglucosan in biomass burning. When we look at the fraction signal of this in these offline aerosol mass spectrometry experiments, what we see is an exponential decrease in this signal. And the exponential decrease that we can see is actually pretty comparable to what was observed for heterogeneous oxidation. We know heterogeneous oxidation can degrade this material, but we wouldn't expect photolysis alone to do that. Now, we may have some chromophoric material that happens to contribute to this peak, right? This is not speciated. But the fact that we have a similar degree of decrease occurring really suggests that we are actually degrading these sugars via direct photolysis in this mixture. So this raises the question of how we're actually doing this. And what I think right now is that we actually have a fair bit of secondary photochemistry that's occurring in these samples. Possibly a lot of photosensitization, a lot of electron transfer, possibly some hydrogen abstraction. And the brown carbon is actually helping to photodegrade and oxidize the other material that we have that would not be necessarily photoactive in these samples. And so that's kind of the big picture questions that I have moving forward for the research that I have. Is I really would like a better understanding of what the chemical composition is of material that we would find in the remote atmosphere, right? Is it largely photorecalcitrant? Or are there some of this photoresistant type materials, things that can absorb solar radiation in the visible region, but are not changing structurally and possibly are able to contribute to this secondary photochemistry that could drive other processes like fragmentation reaction or oxidation reactions in the particles. And so that's kind of a major question that I have for our future work. So conclusions that we have. I started out looking at biogenic secondary organic aerosol. And what we find is there's really big differences between what we see for dry photolysis compared to what's been observed previously for aqueous phase photolysis, really highlighting that we need to understand these photoaging processes under different environmental conditions. We also find for all of the experiments that we're looking at that there's some fast photolysis that occurs, initial rates that are rather rapid, but it's only hitting a small fraction of either the color or the mass in each of these systems. And there's a lot of this photorecalcitrant or photoresistant organic material that is present. And then finally, when we're looking at these biomass burning samples, we're seeing some evidence for secondary photolysis happening in the particles. And this raises the question of whether or not there is photoresistant material in remote organic aerosols that could continue to drive photoaging and could contribute to continued mass loss through photolysis for these organic aerosols in the atmosphere. All right, so I'd like to thank all of the wonderful students who have helped me with these projects, as well as our funding, our collaborators who have assisted us in getting us access to these filter samples, as well as instrumentation, and then you for your time and attention, and I'll be happy to take any questions.
Wonderful. Well, that was quite a lot to take in. So we'll give people just 10 seconds to process it all and see if they have questions to add. See the applause all across the screen. So who would like to start? Questions, comments? I see. John, you got a question.
Yeah. Hi, Rachel.
Hello.
I guess we'll be talking in a few minutes, but I just had one general question. I mean, it was a beautiful talk, really lovely. I mean, you focused, and I totally agree, on the photo bleaching effect at long times. You know, that's sort of going to dominate. But you didn't mention photo enhancement much at shorter times, and that seems to be, you know, Jenny Wong saw that at Georgia Tech, and you know, we've seen it. Other groups have seen it. I think Jamie's group has seen it too. Do you also observe that? Is that something, you know, very short times that you observe?
I have observed it for one of my samples. So one of the aqueous phase samples has a little bit of initial photo browning. The rest of them appear to be photo bleaching fairly consistently. Some of that is coming from the method that I use, right? That MDAE can kind of cancel out some of the browning. If you look at specific wavelengths, you can see the browning occur, right? If you look at like 420, you'll see the browning occur, you'll see the photo bleaching occur at shorter wavelengths in some cases. But if you look at the average across, then you can see a little bit more of an overall photo bleaching. So one of the things we want to try to do is do an intercomparison of the rates at all the different wavelengths that are typically used and compare that to what we have for the MDAE to try to build an understanding of you know, how much of an impact it has, whether you're looking at this on, I think of the single wavelengths as more of a chemical resolved basis as opposed to this MDAE, which is more of like a holistic brown color perspective. So we have one sample that appears to be able to do enough browning that we can actually see it increase and then come back down. It's relatively short, so that browning only happens over the first hour or two, and then it starts its decay. So it's whatever is happening, whether it is I didn't quite get enough scavenger in there and we're getting OH radicals, or if I'm making things that I think I'm actually just making products that have increased absorption at longer wavelengths that's counterbalancing the loss. Those are relatively short-lived, and they are decaying themselves. Yeah. Yeah,
we can talk in a few more minutes, but we have some evidence that looks like it might be radicals like OH radicals, even in the short term, that are doing it. Yeah. Okay.
Yeah, I'm a little bit worried that I need to add more mannitol to try to soak it. Yeah.
Tara.
Hi, Rachel.
Hello.
Lovely talk.
Thank
My question is, I guess, sort of tying the two parts together. In the first one, where you're talking about the recalcitrant, so there, I mean, it seems like everything stops, which suggests not only that these species that are left are not absorbing sunlight, but that they aren't reacting with OH or anything else that might be there.
I only had ozone in the chamber. And if we had, I also had a scavenger in there to try to scavenge my OH radicals. So I was really trying to minimize that.
Well, yeah, no, for sure. I guess my question is, so in the real world, though.
Oh, yes.
Right. So I guess you were just thinking about the photochemical part, not. But okay, so if you have, I'm just trying to think of it all together, because when you have the brown carbon and the other things, it's, yeah.
No, I think we will definitely have OH radical oxidation occurring on those. So you would see a continued decay from the heterogeneous oxidation. And the photo bleaching is then just like that first initial, it's like a punch, and then you got the slow roll from the heterogeneous. What I'm very curious about is whether my additive assumption is correct, or whether heterogeneous oxidation could actually make new chromophores and could allow you to do additional photolysis. So that is kind of an open question. I right now am just kind of combining the lifetimes because that's kind of what we know. But yeah.
Cool, thank you.
Yeah, thank you.
I saw a question from Arthur.
Rachel, thanks for a really nice talk. It's really nice to see all of the all the different techniques that you're using and how you're able to combine them and gain some interesting insights. I have a similar question to what Tara's asking. Do you see like photosensitizing chemistry, where it's like an excited state, and then reacting with things? It might be a little bit complex with a brown carbon, but maybe like in the alpha pinene ozone, like things like that.
I don't think we see photosensitization in alpha pinene, mostly because I haven't really heard of that happening for carbonyls. And so I'd be a little bit surprised. You could potentially have like a radical and hydrogen abstraction thing happening, but we don't really have the types of chemicals that I see as photosensitizers in there, unless something really horrible happened when I generated these samples.
Well, the brown, like the aromatic stuff, those are fair. Like, those are wild, right? They could do all kinds of things, but that might be too complex to see in your system.
Well, I think that that's actually why we're getting so much oxygen incorporated. I think that some of this brown carbon is photoresistant, so it's keeping its brown carbon color, but it's photosensitizing, so it's driving oxidation of the other material in the biomass burning samples. I think that that's partly why we can get so much oxygen, because I don't think should be that much brown carbon to get that amount of oxygen coming in.
Cool, thanks.
Yeah, thank you.
Jen, I think I see your hand up.
Yeah, thanks. Beautiful talk, Rachel. I was curious about when you were discussing the results of the aging experiment in the chamber at MIT, I was wondering whether you need to worry that you have this high offset because you're working at high mass loadings. And so the semi-volatile material is predisposed to stay in the particle phase because you're at a high mass loading, and that might be different in the ambient atmosphere. And then how might that impact photolysis experiments that are done, on bulk systems rather than, do you have to worry that some of these chromophoric materials would actually be evaporating in the real atmosphere?
Yeah, no, that's a great question. We actually did this at different concentrations in the chamber, and I didn't show you all of the data sets. Oh, actually, I guess I did, because there's 3 of them. The one that was the lowest concentration actually had the largest recalcitrant fraction. So it seems to actually get worse and worse the cleaner and cleaner you get. It appears to be the stuff that's semi-volatile that you can get more of into the particles, which kind of makes sense, right? Those are kind of the carbonyl-ish ones. Those are the ones that are more photolabile. And so you see more mass loss when you have a higher concentration of it in the particles. When you have a lower concentration, more of the carbonyls are up in the gas phase, and there's less overall mass loss or a larger photorecalcitrant fraction in the chamber, is what we were seeing. Yeah, it was like, oh gosh, how do we extrapolate this out? Because yeah, that's a real question. Like, is anything photolabile? If you actually go to low enough concentrations. Yeah.
Right. Okay, thanks.
Yeah.
Are there other questions? Let me just check the chat. No, I think we're good there. I have a question, so if nobody else. So I think I missed this at the end. The idea that there is the recalcitrant material. Actually, I should start by saying I'm incredibly impressed that this work has been done primarily by undergraduate students. Is that? Yeah. Okay. So I think that needs to be noted. I think that's impressive all on its own. But to the question, so we have this recalcitrant material. It's no longer subject to photobleaching, but it can induce photochemical reactions that can promote the decay of other compounds, but not of itself. And so I was trying to figure out what is it that's about the property that makes it resistant to also the photooxidation.
Yeah.
And I'm not sure if you covered that or
No, I didn't cover that because I really don't know. I would suspect that some of it is that it's polycyclic aromatic hydrocarbons. And so even if you had a fragmentation event, you've still got a lot of rings. It takes a while to chew through one of those with just photolysis. So I expect that some of it is sort of more complicated, larger molecules, that it's just going to take a while to fully fragment all the way. Some of the other ones probably just have relaxation pathways that have higher quantum yields through non-fragmentation paths. So they will do more hydrogen abstraction, more electron donation, that sort of thing, instead of having an actual bond break in them. And exactly what those are is a really great question, and definitely something that I think needs to be targeted more on a molecular level type study to really understand what's decaying more quickly and what's lasting for longer periods of time, and what is this longer lived material doing in the mixtures.
Yeah. And so I had one big picture question. So if we take your results and we try to extrapolate back, even though there's huge uncertainties in the extrapolation, to the question of forcing, and in terms of the atmospheric lifetime, where does this leave us now? So you had the picture at the start where you had the removal in terms of the lifetime, and you had the removal in terms of photo oxidation. So where do we end up?
Yeah, so I think we need to rethink our photo bleaching, like the total mass loss that can do. I think that assuming that you can get all of this material to photodecay via direct photolysis is an overestimate. And so I think from that perspective, we have the amount of organic aerosol that we have in the atmosphere. We need to figure out how to either make less of it or remove it faster, but we shouldn't be using direct photolysis for that process. The brown carbon side, I think, is a little bit more interesting because that one, you're talking about whether this can do direct absorption or whether it's going to scatter. And for that, I think we need more ambient measurements and a better understanding of what this resistant material is and how much of it we have, and why there's such a big gap between what is observed in India and some of African fires actually appear to be relatively long-lived compared to what we're seeing for these North American fires, which have lifetimes of 12 hours or a day. So there's some real differences potentially in the type of burn or the type of fuel that could be contributing to that. And I think we just need more ambient measurements to try to get a better estimate of the lifetime for the brown carbon that's present. And I know there's people doing that, but that will give us, I think, some better understanding of the direct radiative impact for this system.
Well, thank you.
Yeah, thank you.
I'll check. I know we're just at the hour, so if there's one last pressing question. If not, Jen, I'll throw it back to you to thank Rachel. But thank you, Rachel.
Thank you.
Yeah, thank you so much, Rachel. As Greg mentioned, it's such high quality and quantity of work, not only done by undergrads, but done by undergrads during COVID influenced time. So it's really remarkable. And thanks for sharing your work with us today. Take care.
Thank you so much.
All right. And thank you everyone for coming.